Good afternoon. Today we have to discuss the topic of blotting techniques. Blotting is used in molecular biology for the identification of proteins and nucleic acid and is widely used for diagnostic purposes. This technique immobilizes the molecule of interest on a support which is nitrocellulosic membrane or nylon. It is uses hybridization technique for the identification of specific nucleic acid and genes. The blotting technique is a tool used in the identification of biomolecules such as DNA, mRNA, and protein during different stages of gene expression. Southern blotting was introduced by Edwin Southern in 1975 as a method to detect specific sequences of DNA in DNA samples. The other blotting techniques emerged from this method have been termed as Northern for RNA, Western for proteins, Eastern for post-translational protein modifications. Types of blotting techniques: Southern blotting, Northern blotting, Western blotting, Eastern blotting, dot blot. Dot blot. Southern blot. Southern blot is an analytical technique used in molecular biology, immunogenetics, and other molecular methods to detect or identify DNA of the interest from a mixture of DNA sample or specific base sequences within strand of DNA. The technique was developed by molecular biologist E.M. Southern in 1975 for analyzing the related genes in a DNA restriction fragment and thus named as Southern blotting in his honor. Principles of Southern blot. The process involves the transfer of electrophoresis separated DNA fragments to a carrier membrane which is usually nitrocellulose and the subsequent detection of the target DNA fragment by probe hybridization. Hybridization refers to the process of forming a double-stranded DNA molecules between a single-stranded DNA probe and a single-stranded target DNA. Since the probe and the target DNA are complementary of each other, the reaction is specific, which aids in the detection of specific fragment. Key point: steps involved in Southern blot: extraction and purification of DNA from cells. DNA is first separated from the target cells following standard methods of genomic DNA extraction and then purified. Restriction, digestion or DNA fragmentation. Restriction endonucleases are used to cut high molecular weight DNA strands into smaller fragments. One or more restriction enzymes can be used to achieve such fragments. Separation by electrophoresis. The separation may be done by agrosgel electrophoresis, which the negatively charged DNA fragments move towards the positively charged anode. The distance moved depends upon its size. Denaturation. DNA is then denaturated with a mild alkali such as an alkaline southern of sodium hydroxide. This causes the double-stranded DNA to become single-stranded, making them suitable for hybridization. DNA is then neutralized with a NaCl, sodium chloride, to prevent rehybridization before addition of the probe. Blotting. Denatured fragments are then transferred into a nylon or nitrocellulose filter membrane, which is done by placing the gel on the top of buffer saturated filter paper, then lying nitrocellulose filter membrane on the top of gel. Finally, some dry filter papers are placed on the top of the membrane. Fragments are pulled toward the nitrocellulose filter membrane by capillary action and result in the contact trend of the gel. Baking. Nitrocellulose membrane is removed from the blotting stack and the membrane with the single stranded DNA bands attached on to its back in a vacuum or irregular oven at 80 degrees Celsius 2 to 3 hours or exposed into ultraviolet reaction permanently attach this transfer DNA onto membrane. Hybridization. The membrane is then exposed to the hybridization probe which is single DNA fragment with specific sequences whose presence in target DNA is to be determined. The probe DNA is labeled so that it can be detected usually by incorporating radioactivity of lagging the molecule with a fluorescent chromogenic dye. Washing of unbound probes. After hybridization, the membrane is thoroughly washed with the buffer to remove the probe that is bound non-specifically or any unbound probes is present. Then autoradiograph. The hybridized region are then detected autoradiographically by placing the nucleonitrocellulose membrane in contact with photographic film which shows the hybridized DNA molecules. Applications of Southern blot. Identify specific DNA and DNA sample, preparation of restriction fragment length polymorphism maps, detection of mutations, detection of gene rearrangements in DNA for criminal identification and DNA fingerprinting, detection and identification of transgenic individual, mapping of restriction sites for diagnosis of infectious diseases, prognosis of cancer and prenatal diagnosis of genetic diseases, determination of molecular weight of restriction fragment and to measure relative amounts in different samples. Next one is northern blotting. Northern blot is used in molecular microbiology research to study the gene expression by detection RNA or isolated mRNA in a sample is collected called northern blotting. It is classical method for analyzing the size and steady state levels of a specific RNA in a complex sample. Northern blotting technique is a for detection of specific RNA sequences. Northern blotting was developed by James Alvey and George Strack at Stanford University. Northern blot involves the size separation of RNA in gels like that of DNA because we wish to determine the native size of the RNA transcript. Steps involved in northern blot: RNA isolation, separation by electrophoresis, transfer of RNA to a membrane, hybridization and washing, visualization. 5 steps. RNA isolation. RNA is isolated from the several biological samples. The tissue or culture sample collected is first and homogenized. The samples may be representative of different types of culture for comparison, or it can be for the study of different stages of growth inside the culture. Separation by electrophoresis. Simple or are loaded on gel and the RNA samples are separated according to their size on an agarose gel. RNA samples are most commonly separated agarose gel containing formaldehyde as a denaturating agent for the RNA to limit secondary structure. Denaturing agent disrupts the secondary structure. Separation of RNA is better in glyoxal DMSO system as sharper band for specific RNA detected hybridization. Third step is transfer of RNA to a membrane. Nitrocellulose and nylons are commonly used as membrane. RNA is transferred via capillary or vacuum transfer. Vacuum transfer are more different but requires specific transfer apparatus. Electrophoretic transfer method is available only for nylon membrane due to low concentration of salt required to bind RNA. Hybridization and washing. Hybridization is performed using radio or fluorescently labeled probe to identify specific RNA immobilization. Pre-hydration blocks a single standard probe from binding on non-specific sites on the membrane. Hybridization solution should containing 50% of formaldehyde to ensure hybridization at low temperature and maximize RNA degradation. Membrane is washed in a buffer containing lower concentration of salt to remove excess probes. Visualization, detection of specific transcript through the autoradiography membrane or places over X-ray film. The X-ray films darken while fragments are corresponding to the radioactive probes. Applications. A standard for the study of gene expression and the level of mRNA uh, detection of mRNA transcript size, study RNA degradation, study DNA splicing, study RNA half-life, often used to confirm the check transgenic. Disadvantages: time consuming, DNA degradation risks becomes RNA's contamination in the work environment, relatively expensive for the large scale analysis as huge amount of RNA and reagents records. Again repeating, Western blotting. Western blot is a Western blot is an analytical technique used in molecular biology, immunogenesis, and other molecular biology to detect specific proteins in a sample of tissue, homogeneous or extract. Principle: Western blotting is a rapid and sensitive assay for the detection and characterization of protein. It is based on the principle of immunochromatography, where which proteins are separated into polyacrylate gel according to their molecular weight. The protein thus separated are then transferred or electrotransferred onto nitrocellulose membrane and are detected using specific primary antibody, secondary enzyme and labeled antibody substrates. Steps involved in Western blot: extraction of protein, gel electrophoresis, blocking, treatment with the primary antibodies, treatment with the secondary antibodies, treatment with suitable substrate. Detection can be done by other methods such as colorimetric detection, radioactive detection, chemiluminescent detection, fluorescent detection. Western blotting procedure. Applications: to determine the size and the amount of protein in a given sample. Disease diagnosis: detects antibody against virus or bacteria in serum. Western blotting technique is a confirmatory test for HIV. It detects anti-HIV antibody in patient serum, useful to detect defective proteins, foreign presence disease, defective test for Creutzfeldt-Jacobs disease, Lyme disease, hepatitis B and herpes. Eastern blotting, developed in Thom in 1979. Steps involved in the Eastern blot is a biochemical technique used to analyze protein post-translational modifications such as lipids, phosphomyotics and glycoconjugate. It is most often used to detect carbohydrate epitopes. Dot blot techniques. This technique is used to detect the presence of given sequence RNA or DNA in a non-fractionated DNA sample. The DNA may from many samples can be tested in a single test. Okay. Thank you.